Well, you should have known they were going to have us CERM interns do this video.
You know it was going to happen.
So what kind of stuff do they have you talking about?
Well, I have to introduce stem cells. I have to talk about what they are and where they come from. There's still a lot of misconceptions about these two major topics.
How are you going to address all that?
Well, I need to identify what a stem cell is to begin with. Stem cells and their three major properties, their ability of renewing themselves, the fact that they're unspecialized, they haven't met their fate yet, but they have the potential to become a specific type of cell, which presents a lot of opportunities for different therapeutic purposes. And then I also have to talk about where they come from. A lot of people think that stem cells only come from embryos.
That's wrong, isn't it?
That's very wrong. There's a lot of other sources that people are looking into. One major avenue that people are looking at is induced pluripotent stem cells, called iPS cells.
I think the main focus that I really want to gear my presentation towards is to make sure that people are aware that we're just not running around shooting people up with stem cell lines. There's a lot of work that goes into preparing this research. First, we have to take the cell cultures in the lab and learn how to differentiate them into the cells that we want. The nice thing about stem cells is they can become any other cell in the body. The problem with that is they can become any other cell in the body. So right now we're trying to establish protocols in order to guide the cell down the path of development that we want it at. Once we learn how to do that and make sure we have the cells that we want to use, we can then introduce it into an animal model and see if it has any reactions there. From there, we can start human clinical trials. In a clinical trial, what we're going to have is two different groups, a control group and a test group. The control group's not going to get any test stem cells at all. The test group will get the treatment. We'll then compare the results from the control group to the test group. We'll see if there was any benefit to the treatment, and we'll see if there's any complications.
The purpose of turning this primary cell into a stem cell is so that we can use it to study diseases that affect those multiple organs in the body, and we can use it to test the effectiveness of drugs on those multiple organs of the body, or we can, in the future, use it as transplant tissue, i.e. if you have a bad heart, maybe in the future we can fix that bad heart by injecting some stem cells in it, or creating an artificial organ out of stem cells. There are no approved therapies using pluripotent stem cells, even though we know these cells can give rise to any cell type, and that in theory they should be able to replace any cell that degenerates. The problem is that we've only known about embryonic stem cells or pluripotent stem cells for about the last 10 years. And that's the only work that's been done with them so far has been in the lab. There's been no work done on people. So now we turn to people who are desperate now with diseases that bone marrow transplants, for example, won't help. What do they do? And what do you do if you have a family member who has some really terrible disease, or even a child with autism, which is terrible to live with? And then you look on the web, you screen, you Google autism and stem cells, and there'll be a list of 10 places that will offer you a therapy, a stem cell therapy for autism for your child. What do? If you look at those websites, you'll see that the people who offer those services are sometimes based in the U.S., but that the treatment itself, the stem cell transplant, will always take place in some other country.
Outreach is a combination of things. I think we need to get out into the sectors and the population in the community that find us so controversial. Ask them what their questions are, what their concerns are, and outline them. Let them know how far that we have come in technology based on the research that we have done. Also to let them know that it's very important that we have a dichotomy of people, a wide range of diverse cultures. We can't have one control type that is not going to be indicative of every single ethnic group that this world supports. We are a global world. We are all interconnected at a cellular level. And so with that, outreaching to these individuals, letting them know and educating them. There has been a lot of harm done in the name of science in the past. And people have long memories, and they don't forget that. We need to be more sensitized and not desensitized. We can't just continue with our work without the support of other organizations.
I'm a high school student right now. I'm 17 years old and I'm in my senior year in high school. And I've been working here since October of 2009. It was a huge joy for me to start working here. I applied of my own will. I was looking out for places that were willing to hire an intern like me because the experiences that I would get from working in a lab and just being able to work with my hands with the same, materials and have the same, chances to work with doctors as most students like me will never have. It was just amazing.
It was a pleasure for me to work with Gabriel. He's one of the best interns I've ever had. And for a high school student, it was much better than I ever thought. And the projects that we're actually working on right now are making iPS cells from different patients. In this case, amyloid lateral sclerosis, Lou Gehrig's disease patients. And also we're working with normal human embryonic stem cells. And we want to differentiate them into neurons and then study what happens to the cells when you make neurons out of them. So those are two current projects that we're working on, and Gary has been very helpful in getting those projects to succeed.
Let me give you a word about clinical trials. If somebody says that something is a clinical trial, that means the patient does not pay for it. If somebody says that they're running a clinical trial and they want you to give them $25,000 in order to enroll in it, then it's not a clinical trial. It's just a way of getting money from you. So clinical trials they may seem kludgy. The FDA may seem like it doesn't move very quickly, but there have been so many examples in which drugs and therapies have actually been marketed. They've been put on the market, and then they've caused a lot of harm to people. So the idea behind a clinical trial is to anticipate the problems that might arise.
Connected at a cellular level, once upon a time, 3 friends decided to contribute to the betterment of humankind. They donated a biopsy to stem cell research. It was an ethnic blend because there's still a lot of cultures that are hesitant. Pluripotent, regenerative cells splitting and splicing, living forever inside of a petri dish. They agreed that this would be a bonding experience. What a novel idea to reproduce over and over and over again under the lens of a microscope, locked inside of this glass dish. While scientists search feverishly for a cure for the degenerated heart, a weakened lung, a cancer patient who's scared and very young, a single cell is extracted and reduced down to its basic form, reproducing, genetically re-engineered, reincarnated into the muscle of a heart, the tissue of a lung, vessels through a kidney, potential for a spinal cord's victim mobility. Countless hours of analysis, observation, theory, hypothesis, and documentation. Restricted and limited funding, alongside of the skeptics humming, will not stop the scientists. Gobbled up at the bottom of the food chain, trust me, if we remain the same. Afraid of technology and science, we must be like the stem cell we seek, extracted, allowed to reproduce, cure, heal, and change.